Welcome to this lecture on the structure of DNA. At the end of this session, you will be able to understand the discovery of DNA. You will be able to illustrate the structure of DNA, and also you will be able to describe the complementary base pairing of DNA. So first of all, we'll learn the overview of DNA. DNA stands for deoxyribonucleic acid. This chemical substance is present in the nucleus of all the cells in all living organisms. DNA controls all the chemical changes which takes place in the cells. This is the chemical basis of heredity organized into genes, the fundamental units of genetic information. Therefore, it is regarded as the reserve bank of genetic information. In simple, we can say as genes are like the story and DNA is the language that the story is written in. When we talk about the discovery of DNA, it starts in the year 1928. Frederick Griffith, he is a bacteriologist. He discovers that a factor in diseased bacteria can transform harmless bacteria into deadly bacteria. In the year 1944 to 1950, Erwin Chargaff discovered that DNA is responsible for heredity and that it varies between species. In the year 1952, Rosalind Franklin discovered the X-ray photo of DNA. She also established the helical conformation of DNA. In the year 1953, Watson and Crick described the molecular structure of DNA. With the help of Rosalind Franklin's X-ray crystallography, they both discovered or proposed the molecular structure of DNA. And in the year 1962, they were awarded with Nobel Prize for their discoveries. And now let us learn the structure of DNA. DNA, it is a right-handed double helical structure. It consists of two strands. So in the picture you can see there are two strands and these two strands are called polyribonucleotide chains. They are twisted around each other on a common axis. On a common axis. The two strands are anti-parallel in direction. That is, one strand it runs from 5 prime to 3 prime and the other strand it runs from 3 prime to 5 prime. Hence, the direction is anti-parallel in direction. Composition of DNA. This DNA, it consists of a sugar molecule and the sugar that is present in DNA is called as deoxyribose. It contains a phosphate molecule and it contains nitrogenous bases. When we talk about nitrogenous bases, there are four different types of nitrogenous bases. They are guanine, cytosine, thymine and adenine. So these are nothing but the purines and pyrimidines. Again, when we talk in detail about the structure of DNA, the width or the diameter of a double helical structure is 20 Armstrong, which is also, we can also say as 2 nanometer. Each turn of the helix is 34 Armstrong, that is 3.4 nanometer, with 10 pairs of nucleotides. Each pair placed at a distance of about 3.4 Armstrong. Each strand of DNA has a hydrophilic deoxyribose phosphate backbone, that is 3 prime to 5 prime phosphodiester bonds on the outside of the molecule, while hydrophobic bases are stacked inside. The two polynucleotide chains are not identical but complementary to each other due to base pairing. So here they are not identical. These two strands are not at all identical, but they have complementary pairs. And these two strands were held together with the help of two different bonds. One is the hydrogen bond, the other one is phosphodiester bond. The hydrogen bonds formed by the complementary base pairs. The hydrogen bonds are formed between a purine and a pyrimidine molecule, pyrimidine base. The phosphodiester bonds form between sugar and phosphate, which forms the backbone of nucleic acids. Next comes the complementary base pairing of DNA. The adenine and thymine pair has two hydrogen bonds, whereas the guanine and cytosine pair has three hydrogen bonds. The guanine-cytosine pair is stronger by about 50% than adenine and thymine pair. The complementary base pairing in DNA helix proves the Charhaff's rule. The content of adenine equals to that of thymine and guanine equals to that of cytosine. So here you can see in the picture adenine paired with thymine with the help of two hydrogen bonds, whereas the guanine it pairs with cytosine with the help of three hydrogen bonds. So this is called as complementary base pairing of DNA structure. So in today's lecture, we have learned about the overview of DNA structure, the discovery of DNA, and you have also learned about the complementary base pairing of DNA. I end this lecture by saying a short poem on DNA, and this is the one. Life instructor, bodybuilder, color constructor, shape sculpture, plant grower, triad tailor, constant creator, similarity shaper, difference decider. I am the DNA. Thanks for watching.